Hello guys, good day. I am Miss Merle, and for today we will be having the discussion about the non-fermentative gram-negative bacilli. So for the objectives of this discussion, we have nine objectives. So the first one is we need to describe the general characteristics of non-fermentative gram-negative rots. Second, compare the metabolic pathways used by non-fermentative organisms. Third is discuss the natural habitat of the non-fermentative gram-negative bacilli. Fourth is describe the types of infections that non-fermentative organisms cause in patients at high risk for infections. Fifth is name the most clinically significant non-fermentative gram-negative bacilli. Six, discuss the incidence of infections caused by various non-fermenters in patients with cystic fibrosis. Recognize the initial clues to non-fermentative organisms isolated in clinical specimens. 8 is describe the typical biochemical reactions and characteristic features of the most commonly encountered non-fermentative organisms. And 9 is the last one is to evaluate the typical antimicrobial susceptibility pattern of most commonly encountered non-fermentative organisms. So for the outline, we have two headings. We have the general characteristics of the non-fermenters, in which we will be talking about clinical infections and biochemical characteristics and ID. The second heading will be about clinically significant non-fermentative gram-negative bacilli. So we will be talking only about the clinically significant ones. We have the under this category, we have the Genesis Pseudomonas fluorescent group. We have the Pseudomonas non-fluorescent group, Acinetobacter, Steno trophomonas maltophilia. We have the Burkholderia, Moraxella, Oligella, and Psychobacter. So basically, gram-negative bacilli are divided into aerobic. We have the aerobic, and we also have the obligate anaerobic bacilli. Aerobic gram-negative bacilli can be further divided into at least two large groups: those that ferment carbohydrates, called fermentative or fermenters, and those that do not ferment carbohydrates, called non-fermentative or non-fermenters. This presentation will discuss the miscellaneous non-fermentative organisms that have become clinically significant because of the increasing numbers of immunocompromised patients. This group of patients often have multiple risk factors for infection with these normally uncommon pathogens. The groups of organisms discussed in this chapter, as what I have mentioned previously, include Pseudomonas, Acinetobacter, we have Steno and other glucose-negative oxidizers, and species of gram-negative bacilli that fail to metabolize carbohydrates. So we have the general characteristics of non-fermenters, and these are non-fermenting gram-negative bacilli are grouped together. Why? Because they fail. Take note, they fail to acidify an oxidative fermentative OF medium when it is overlaid with mineral oil. So take note of the mineral oil overlay. And second is they fail to acidify triple sugar iron agar or TSIA buds. And they prefer and grow much better in an aerobic environment. But some do not grow in an anaerobic environment at all, and some group members oxidize carbohydrates to derive energy for their metabolism, and these are referred to as oxidizers, and others do not break down carbohydrates at all, and they are referred to as non-oxidizers or acyclolytic. We also have the general characteristics of non-fermenters, and most are oxidase positive, and they are ubiquitous in the environment and found in most environments, typically in soil and in water, on plants and decaying vegetation, and in many foodstuffs. They prefer moist environments, and in hospitals they can be isolated from nebulizers, dialysate fluid, saline, catheters, and other devices. Non-fermenters can withstand treatment with chlorhexidine and quaternary ammonium compounds. They are rarely, if ever, part of the normal host microbiota, but can easily colonize hospitalized patients. So it's a very bad thing. It's easy for them to colonize immunocompromised patients, and many of the non-fermentative gram-negative bacilli tend to be resistant to multiple classes of antimicrobial agents. And non-fermenters are responsible for a number of serious infections, including septicemia, meningitis, osteomyelitis, and wound infections, often following surgery, after surgery, or post-traumatic events. Usually, the individual who contracts an infection with one of these organisms is hospitalized, or those who have been recently discharged from the hospital. And for the risk factors for diseases caused by non-fermentative gram-negative bacilli, we have four major groups of risk factors. We have immunosuppression, trauma, foreign body implantation, and infused fluids. So those patients having DM, those having CA, taking having steroidal medication regimens, and those who were able to undergo transplantation are having those risk factors too. And we have knife wounds, puncture wounds, and gunshot wounds, and of course, including surgery and burns. We also have foreign body implantation, so foreign objects like prosthetic devices, implants, contact lenses, and catheters. And also for infused fluids like as simple as saline irrigations and dialysate fluids can become risk factors of contracting these organisms, contracting the infection from these organisms. So the biochemical characteristics and identification. So here I'm trying to differentiate. We have the fermenters and the non-fermenters. And since we are talking about the non-fermenters, so since they are called non-fermenters, they really fail to ferment carbohydrates, and there is the absence of acidic reactions in the anaerobic portion of the DSIA or KIA. And then in contrast, we have the fermenters. They are positive for the presence of yellow buds, so that's acidic, and also positive for an acid or alkaline slant, the slant. So here we're talking about the buds and the slant on DSIA or KIA within 18 hours of incubation. So the true fermenters, they are really fastidious, and they do not easily acidify the butt or slant of ATSIA like other fermenters, but do show reactions if more enriched media are utilized. So for the some characteristic clues can indicate or signify the presence of a non-fermenter. So how can we know if a non-fermenter is there? So we have first one is positive for the oxidase reaction, although reaction can be weak and variable. We also have non-reactivity in 24 hours in commercial multitask kit systems, used primarily or mainly for the identification of Enterobacteriaceae, and then absence of acid production. There is no acid production in the slant or butts of TSIA or KIA, and resistance to a variety of classes of antimicrobial agents like aminoglycosides, third generation cephalosporins, and penicillins, and fluoroquinolones. And we have many classification systems have been devised for grouping non-fermenters, and one system uses the reactions of three common tests. So we have the growth and MAC agar, oxidase reaction, and the glucose OF test. And the four groups of non-fermenters that make up most isolates routinely seen in the clinical labs today, we have Pseudomonas aeruginosa, Acinetobacter, Burkholderia, and Steno trophomonas maltophilia. So for the decision to identify many of the non-fermenters is based on the site from which they have been isolated. That is, whether they have been recovered from a normally sterile site in pure culture or from a non-sterile site in which 3 or 4 other bacterial species are also present. So in the former case, this one, it may be decided that definitive identification and susceptibility testing are needed or required. While in the non-sterile site, Genus identification might be appropriate, which can be achieved through the use of few biochemical tests like oxidase, growth on MAC, glucose utilization, motility, and indole. And alternatively, we have identification as a non-fermenter, not P. aeruginosa, Acinetobacter, Burkholderia, or as maltophilia, may be all that is needed, especially when cultures are mixed with many other bacterial species. So here we have the common characteristics to the non-fermenter groups. So we can base it on pigmentation. So we have those which are producing yellow pigments, pink pigments, the purple ones, blue-green ones, violet, lavender, and tan. And we also have based on wrinkling on the colonies, we have the odor. So if it's sweet, it will signify these organisms. If the odor is reminiscent of popcorns, then we can think of Neisseria. And then if they are non-motile, there's a lot of them. And for those which are negative for oxidase, and we also have this one which is positive for hydrogen sulfide production. So right now, this is the latest trend in the diagnosis of microorganisms, not only for bacteria but also for fungi and viruses. We have MALDI-TOF, a mass spectrometry. This is a method for the identification of bacteria and fungi from colonies based on the analysis of the unique spectra of their peptides. So peptide spectrum when separated in a mass spectrophotometer. So identification can be made within minutes, and it provides high-level accuracy and reproducibility if compared with the gold standard of molecular sequencing. Before, the gold standard is this one, but it has a higher level of reproducibility or accuracy than molecular sequencing. And then the genus Pseudomonas accounts for the largest percentage of all non-fermenters isolated from clinical specimens. Characteristics common to most of the Pseudomonas, including members of the genus Pseudomonas and several Pseudomonas-like organisms, include gram-negative bacillus or cocobacillus, strictly aerobic. They are motile because they have this polar flagellum or polar tuft of flagella. They are oxidase and catalase positive. Often they grow on MAC agar, and usually they are an oxidizer of the following of carbohydrates, but some species, take note, are asaccharolytic. And let's start with the Pseudomonas fluorescent group. Under it is the Pseudomonas aeruginosa. This one is the most commonly isolated species. Of the genus in clinical specimens. It can be found in moist environments. Again, moist environments like hot tubs, catheters, pools, and hospital humidifiers. Reservoirs for P. aeruginosa include plants, soil, and tap water. And it is an uncommon part of the normal bacterial microbiota and is isolated from less than 12% of normal stool specimens. Well, this one, take note, if you are a healthy person, then it can cause mild illness. However, if you are immunocompromised, it can indeed cause severe infections, and may however account for 5 to 15% of all nosocomial infections, especially pneumonia and bacteremia. Pseudomonas aeruginosa is the leading cause. It is the leading cause of nosocomial or hospital-derived or hospital-acquired respiratory tract infections, and it accounts for up to 6% of all the bacteremias and has as many as 75% of nosocomial bacteremias, and it is the third most common cause of gram-negative bacillary bacteremia after Klebsiella pneumoniae and E. coli. And this one is a common cause of lung infections in people suffering from cystic fibrosis. So, Pseudomonas aeruginosa, when this organism is isolated from a sterile body site like pleural fluid, joint fluids, tissues, or blood, or in CSF, it almost always constitute a true infection. So it's a true infection when we can isolate this one from a sterile body site. And aeruginosa can colonize mucosal surfaces such as the oropharynx, and patients in the ICU who are mechanically ventilated may quickly become colonized with P. aeruginosa. And we also have other less serious conditions, less serious, but still, there are still infections. So we have otitis externa in divers or swimmers, and this is now a necrotizing rash referred to as jacuzzi or hot tub syndrome. And also, this organism can cause infections in artificial nails, in the nail beds in artificial nails. It's not a good thing if you have artificial nails, and then this organism will invade your nail beds. Then that's what we call Pseudomonas aeruginosa. So the virulence factor. So the thing, it has a lot of it can produce a variety of factors that can lend to its pathogenicity, such as endotoxin or lipopolysaccharide or the LPS. This endotoxin, the LPS. We have the pili, capsule flagella, phospholipases, type 3 secretion system, and several exotoxins, the proteases. These exotoxins and the proteases, hemolysins, lecithinase, elastase, and deoxyribonucleases. But the most important exotoxin should be, we should not forget that it's exotoxin A because it functions similar to the diphtheria toxin. It blocks a protein synthesis. So that's the thing. Exotoxin A, aside from being cytotoxic, it blocks protein synthesis. Okay. A majority of the virulence factors function against phagocytic activity, take note. And they are also largely cytotoxic in nature. So we have the identifying characteristics. So members of the Pseudomonas fluorescent group, which includes the species on aeruginosa, fluorescens, putida, veronii, bacilli, and montillii, produce a pyoverdin. What is pyoverdin? It is a yellow-green or yellow-brown pigment. It is water-soluble and fluoresces under short wavelength UV light, and most of the strains of Pseudomonas aeruginosa will also produce the blue water soluble pigment pyocyanin. Pyocyanin combined with pyoverdin produces the green color, which is characteristic of Pseudomonas aeruginosa colonies on Mueller-Hinton agar. So the very pretty green color colonies on MHA. This one. And other soluble water pigments occasionally produced by Pseudomonas aeruginosa. We have a pyorubin, so rubin is red, and we also have pyomelanin, it could be brown or black. Verdin is green, cyanin is blue. We also have Pseudomonas aeruginosa, the treatment. So I tried to make it easy for you. So they are resistant. These organism is resistant to the following and is susceptible. So do not give this to the patient. Rather, give these medications to your patients after after a thorough laboratory um, checkup. And then we also have severe P. aeruginosa infections need a combination therapy of the following for severe types. But if the infection is mild, then there's no need for a combination therapy. But if it's getting severe, then that's the time that we need to have the combination therapy. Also, we have the Pseudomonas fluorescens and Pseudomonas putida. The good thing with these, they are of very low virulence, very low, rarely causing clinical disease. They have been isolated from respiratory specimens and contaminated blood products, cosmetics, urine, hospital equipment, and fluids, and both have been documented, although rarely as causes of UTIs and post-surgical abscesses, emphysema, septic arthritis, and other wound infections. And P. putida has been associated with catheter-related sepsis in patients with cancer. So this one. Isolation of P. fluorescens from blood culture bottles in asymptomatic patients has been responsible for the clusters of pseudobacteremia, probably related to contaminated catheters and catheter-related devices. Both species can grow at 4 degrees Celsius and have been linked to transfusion-associated septicemia. And both produce pyoverdin, the green pigment, but neither produces pyocyanin. So they produce pyoverdin but cannot produce pyocyanin, and then or they cannot grow at 42 degrees Celsius. Growth at 4 degrees, they love the cold, but at higher temperatures they cannot. And then the key characteristics of P. aeruginosa, fluorescens, and putida, they cannot reduce nitrate to nitrogen gas, but yes, they can produce acid from xylose. And then what else? Gelatin hydrolysis can be used to differentiate the two species from each other. So putida is negative while fluorescence is positive. So this one is the positive, and for the negative, this one, there's no hydrolysis. Next, we have the Pseudomonas statzeri. Although a rare isolate, it's very rare, an even rarer pathogen in the clinical lab, it is usually recognizable because of its characteristic macroscopic appearance of wrinkled, leathery adherent colonies. So this one, wrinkled, leathery adherent colonies that may produce a light yellow or brown pigment. Isolates are ADH negative and starch hydrolysis positive, the combination of which is specifically distinguishes P. statzeri from most other Pseudomonas species. And P. statzeri is a soil denitrifier, so meaning it can grow in an anaerobic environment in nitrate-containing media producing nitrogen gas. So it can produce nitrogen gas. We also have Pseudomonas mendocina. We can see or find this one in soil and water, and rarely isolated from human specimens, often considered a contaminant in the lab. And this one, in contrast, does not produce wrinkled, but rather produce non-wrinkled flat colonies, which are yellowish-brown pigment. And however, pigmentation is variable, but if there is a pigmentation produced, then it is having a smooth, buttery appearance. This one is motile because it has a single polar flagellum. And then we also have Pseudomonas pseudo-alkaligenes and Pseudomonas alkaligenes, often considered still contaminants. This one is a contaminant, right? Mendocina, this one is also a contaminant. They are oxidase positive and biochemically negative in them. Several tests for which other Pseudomonas species test positive, and they can grow on mock agar, and the reduction of nitrates to nitrites or nitrogen gas is quite variable. They are motile by means of a polar flagellum, so it's flagellated. And for us to have a definitive species differentiation, we need to have MALDI-TOF for this or DNA sequencing. And to differentiate the pseudo-alkali genes and the Pseudomonas alkali genes, so we have ADH positive for ADH production for pseudo, and a weak fructose fermentation, but this one it is negative and we cannot tell. And also we have the Pseudomonas luteola and Pseudomonas oryzihabitans. Habitat unknown, rarely isolated from humans, but from wounds, blood cultures, abscesses, peritoneal and chronic ambulatory peritoneal dialysis, these organisms were isolated. They are of course gram negative, non-fermentative, oxidase negative, catalase positive, motile. Most of them are motile, and oxidize glucose. They can grow on MAC and often produce a non-diffusible yellow pigment. And take note, wrinkled or rough colonies after 48 hours of incubation. And for Pseudomonas or oryzihabitans, it has been found in Japanese rice paddies and been isolated from hospital drains and respiratory therapy equipment. So for Acinetobacter, only 11 species have been officially named. The two species most commonly seen in the clinical specimens are Acinetobacter baumannii, the glucose oxidizing non-hemolytic strain, and we also have Acinetobacter woofy, the glucose negative non-hemolytic strain. So we have two species commonly seen. Again, A. bone manii, and we have woofy or luffy. It's up to you. So they're both non-hemolytic, but one can oxidize glucose, the other one cannot. And then 25% of the adults carry the organisms in their skin, and 7% carry these organisms in their pharynx. And then 45% of the patients with a tracheostomy may be colonized. So those patients with a tracheostomy are really at risk to develop infections from these genus. And ventilator assisted pneumonia and sepsis due to a baumannii have a high mortality rate. So take note, it's really deadly, highly fatal. We also have this one for the clinical infections caused. Well, these organisms are opportunists. They primarily mainly affect patients with weak immune systems and those having underlying disease conditions or coexisting comorbidities. Usually they occur in areas with high fluid content, so in the urinary tract, in the peritoneal fluid, and in the respiratory tract. And then, however, this species is much less virulent when isolated. And when isolated, usually it indicates or signifies contamination or colonization rather than infection. We also have the how to identify the species under Acinetobacter. They are strictly aerobic. They appear as gram-negative coccobacilli or even gram-negative cocci on gram stain. And Acinetobacter organisms can resist decolorization, and retain the crystal or CV stain, which often leads to misidentification. And they are gram-positive cocci. And smears made from blood culture bottles, and they are oxidase negative, catalase positive, and non-motile. They can, much of them can grow on laboratory media, including mac agar, and some species. This one is this is the most important. They produce a purplish hue that resemble that of a lactose fermenting bacterium. So the purplish hue, okay. Next, we have Steno, trophomonas, maltophilia. This is the third most common non-fermentative gram-negative bacillus. And again, widespread in the environment, ubiquitous. And the isolates are, again, ubiquitous in the environment and being found in water, sewage, and blend materials. They're very common in the hospital setting where they can be found contaminating blood drawing equipment, disinfectants, transducers, and other equipment. So, like, they're not really that good after all. And we also have the clinical infections caused by these species. They are regarded as a saprophyte or colonizer. They can easily invade and colonize the respiratory tract of his hospitalized patients. And the most important risk factors in affected individuals were we have immunosuppression and hospitalization, especially in an ICU, in the presence of a CVC or central venous catheter. And identifying characteristic of Stenotrophomonas maltophilia include they are gram-negative bacillus, oxidase negative, non-fermentative, and this one, bluish colonies on mac agar. That's so blue after, but they tend to appear bluish. The colonies tend to appear bluish, and they're catalase positive. So we have trimethoprim, sulfamethoxazole, That's the drug of choice for most infections with Steno trophomonas maltophilia. Sorry. We also have the Burkholderia cepacia complex. It's a complex, so at least it has 18 distinct genomic species or genomores, and contains plant pathogens that have arisen as opportunistic organisms, usually correlated associated with pneumonia in patients with cystic fibrosis or CGD or chronic granulomatous disease. And these organisms can cause endocarditis, especially in IV drug users, pneumonitis, osteomyelitis, UTIs, and dermatitis, and other wound infections resulting from the use of contaminated water. So it's not. We should be very careful with when using water. If we don't know the source or whatever, then refrain from using such. And such organisms are isolated from fluids used in the hospital, like IV fluids and irrigation fluids, anesthetics, detergents, disinfectants, and nebulizers. And they grow well on most lab media. However, they may lose viability on SBA in 3 to 4 days without transfers, and the selective media, if using the selective media with antimicrobials to reduce the growth of P. aeruginosa. So we have the following selective media. And this one, the B. cepacia selective agar BCSA is considered as the most effective in reducing or decreasing overgrowth while maintaining good recovery of B. cepacia, in wherein we can observe non-wrinkled colonies. And we also have Burkholderia Burkholderia which causes glanders. It is a respiratory tract zoonosis affecting animals, primarily livestock like horses, donkeys, and mules. And this one is considered a potential bioterrorism agent. So the occurrence should be reported to the local or state public health departments. And susceptibility tests are performed only in approved labs since this is considered as a potential bioterrorism agent. And while this organism is host-adapted and does not grow in the environment, this one is a non-motile gram-negative coccobacillus in non-pigmented colonies in 2 days. And as what the CLSI recommended, broth microdilution with Brucella broth must be utilized for its identification. And we also have, if we had glanders with Burkholderia Burkholderia we also have melioidosis with or Whitmore's disease in Burkholderia pseudomallei. So we have Malay. So melioidosis. What's that one? What is it? Sorry. It is an aggressive granulomatous disease or pulmonary disease caused by the inoculation, inhalation, ingestion of the organisms with metastatic abscess formation in the lungs, and other viscera. And the presence of this disease always correlates with an overwhelming septicemia, so it's deadly again. Most common presentation would be orbital cellulitis, the cryocystitis, and draining abscesses in the skin. And this one is found in the water and muddy soils. Common in Southeast Asia, including Vietnam and Thailand, in Northern Australia and Mexico. So this one, pseudomallei, is a non-fermentative. It can produce non-fermentative wrinkled colonies. Wrinkled, take note, in sheep's blood agar. It utilizes lactose, and this one, it has a very specific selective medium, ash-down medium, which is supplemented with colistin. And characteristically, this organism produces deep pink colonies because of the absorption of neutral red in the medium, and also it can produce or give off an earthy odor aside from the deep pink colonies in the ash down medium. And for us to identify, we have two ways, MALDI-TOF and with the multiplex PCR assays. MALDI-TOF for the rapid confirmation of Burkholderia pseudomallei septicemia, and this provides really real definitive identification. And for this one, for the multiplex PCR, it's for the ID of many Burkholderia species, including cepacia complex and pseudomallei. Well, this one can give you a rapid and accurate laboratory diagnosis. And we're close to the end. We have Moraxella, Oligella, and Cepacia. These are the members of the family Moraxellaceae. They are non-motile, strongly oxidase positive, gram-negative cocobacillary to bacillary organisms. And then they are in general biochemically inert with regard to carbohydrate oxidation, meaning they are asaccharolytic. What do you mean by asaccharolytic? They are like biochemically inert with regard to carbohydrate oxidation, and they are strictly aerobic. They really need oxygen and usually susceptible to penicillin, an unusual characteristic for this non-fermenter. And these isolates are opportunists that reside on the mucous membranes of humans and lower animals and can be isolated from where respiratory tract, urinary tract, and the eyes. However, they rarely cause disease in humans with the exception of M. catarrhalis. So M. catarrhalis is the most frequent isolate in the genus Moraxella. So M. catarrhalis. So we also have M. non-liquefaciens is the second most commonly isolated member of the genus, can be found in the respiratory tract, but the good thing, it rarely causes disease in humans. And the last one, we have Oligella, two species, Erythralis and Eurolytica. They are small, paired, gram-negative bacilli or coccoid organisms, often isolated from the urinary tract, and they do not grow on mac agar. They are non-oxidative. They are positive for PDA and oxidase, and also nitrate and nitrite with gas formation, and they are susceptible to penicillin. To differentiate Eurolytica and Erythralis, we have this one is motile. Erythralis is not motile because Eurolytica has a peritrichous flagella, and it has been found that Oligella Eurolytica causes bacteremia in AIDS patients, and for Erythralis, from the word urethra, so we can correlate this with this one is a commensal organism in the genitourinary tract. And that's the end of my discussion, and I hope you have learned something from it.